cloning. Edexcel IGCSE biology. Now, micropropagation. Ever heard of the term micropropagation before? What was it?
Mm,
Tissue culture is
I
a
don't remember.
which very small micro pieces of plants and tissues are grown and cultured using nutrient media. Because they are initially grown in petri dishes or nutrient, we say that they are grown in vitro outside a living organism. You take microscopic pieces of plant tissue, grow them in a culture media, and grow them inside petri dishes, inside a medium, and that medium can contain a lot of nutrients and different, maybe plant hormones as well outside a living organisms. Cells are scraped from the
Yes.
parent plant. You take a parent plant and scrape some cells off it. These are known as explants. The pieces, the small pieces taken from parent plant, scraped with a knife. The surface of the explants are sterilized using a disinfectant followed by a rinse with sterile water, and sterilized explants are then transferred to a sterile petri dish containing the sterile nutrient agar as well. Now, first they are sterilized with disinfectant, rinsed with sterile water, transferred to sterile petri dish. What does the term sterile mean? Yes, tell me quick.
So that it is not infected, it's very clean and has
Pure,
no,
unreactive. The growth medium
yes, no
that
bacteria.
we place the explants inside, we place these explants inside a growth medium in a petri dish. Okay. Now these are the pieces and they are inside a medium. This medium, the growth medium encourages the explant cells to grow and divide into small masses of cells known as calyces. Each callus is transferred to a fresh growth medium that contains a range of plant growth regulators hormones, and the presence of these hormones causes the callus to develop roots, stems, leaves, forming a whole plantlet. Plantlets can be transferred to individual potting trays and then they can develop into plant as well. Now, first, what was the very first step of micropropagation? Can you tell me?
Uh, you you take this piece of the parent plant, the explant, and then
Plants, any
you sterilize.
pieces. Okay.
Yes, and then you put them, and then you sterilize them with the
Hmm.
disinfectant,
Yes. Then put them
and
where?
then with like pure like sterilized water, and
Hmm.
then you put it to like the petri dish, sterile petri dish,
Which has
and
what?
then a nutrient agar.
The growth medium. Okay, which contains enzymes, regulators, hormones, and then they develop these explants into what? Calyces.
Yes,
These calyces then slowly grow what? Roots, and then finally they grow in stems as well, and then finally these
yes,
calyces grow into a full plantlet as well. So first of all, explants are taken using sterilized tools, disinfected and rinsed in sterile water. Then they are placed in a sterile solid medium. Now this yellow thing, what does this contain? Proteins, regulators, hormones, nutrients, okay, enzymes maybe. And this cotton
mm-hmm.
wool prevents the bacteria entering but allows for gas exchange. Now slowly these calyces start to form and the roots as well. And then these calyces are transferred to a medium with plant growth regulators. पहले जो medium है ये medium बिल्कुल आपके पास एक sterile solid medium है. But once they grow into calyces, you have to grow shoots, right? So now you move it into
Yes,
a second medium, and this new medium contains what? Plant growth regulators such as auxin, such as gibberellin, such as many other. So these calyces in the second medium, this was the first sterile medium, अब ये second मीडियम है, इसमें सिर्फ छोटे बहुत न्यूट्रिएंट्स थे. यहाँ पे प्रॉपर प्लांट ग्रो रेगुलेटर्स हैं, एंड नाउ दे आर गोइंग टू ग्रो स्टेम एज वेल, लीव्स एज वेल. वी कॉल इट अ totally cloned plantlets, and they are then transferred to the soil in pots before planting outside as well, so you can first take some proper care. So what are the advantages of micropropagation? Can you tell me any idea?
so they are exactly genetically identical to the parent plant, and so it can be used to like commercial uses, like if you have like tomatoes and stuff and
Good.
plants.
Amazing. Clones are genetically identical individuals. The cloning of plants has many important commercial uses. For example, it allows a variety of plant with desirable characteristics to be produced cheaply, a greater yield, and quickly as well. It can be grown at any time of the year. They are all identical to each other. They are all disease-free or resistant plants. And you can also ensure that the diseases prevalent in other areas of the world are not imported and spread by ensuring native varieties of plants are produced. Native, what does the native varieties means? Promoting indigenous or native varieties. What does that mean? Native versus imported. Imported होते हैं foreign species of plants. Imported, or you can say non-native. Non-native is your own country's plant which are locally
Yes.
produced, which are locally adopted. ठीक है? Native indigenous
Yes.
plants, so you have to promote their varieties rather than importing other. For example, in Islamabad in 1970s, what happened that we tried importing some other trees, and now you can see every year we have those pollen allergies. Why is that? They're not adopted to our environment. We have just
Yes.
bought them
So
from other countries just for beauty purposes, ornamental plants.
yes.
If so, we should promote our own native plants, indigenous plants.
Yes.
Without so you can produce large enough quantities to supply the demand in one country without importing plants abroad. So you should produce more varieties of your own plants. Tissue culture can also be an important process in preserving rare plant species. But what are the disadvantages? Trained personnel is not available in every country who are so much trained and have all these skills of micropropagation. Plus, you need a sterile laboratory as well. Then also all the plants produced are genetically identical, and so will all the vulnerable to the same species and pests as well. The lack of genetic variation would make them less able to adapt to environmental change as well. Is it clear? What is
Yep.
micropropagation? Good. So can you tell me all
And
the
my
steps quick?
so you start by taking the parent plant
Hmm.
and then you put them and sterilize them and put them on a with sterilized water and put them on the sterile petri dish
Okay.
containing the nutrient agar, and then you put them into uh, grow uh, growth mediums,
Good.
and then you allow them to get like the small little sprout, I guess.
Mm-hmm.
And then after they have done that, you put them in a bigger medium with plant growth hormones,
Regulators.
and
Yeah.
then you put them in soil before you
Exactly.
plant them.
Amazing.
If you already have an animal or plant with all the characteristics you want, then it would be ideal just to make loads and loads of identical copies of it. That is what cloning is, making genetically identical copies of organisms. This is done naturally in nature by organisms that do asexual reproduction, by mitosis. That produces genetically identical organisms. You only need one parent, there are no gametes involved, there is no mixing of genetic information, and all the offspring are genetically identical to each other. So that is a type of cloning, asexual reproduction. Another type of cloning that's done very easily is taking cuttings from plants. It's been done for hundreds of years. Gardeners take cut off a stem from a plant that they particularly like, and they stick that stem of the plant in the ground, and it will grow into the same as the parent plant. It's a type of cloning. Plants, though, can be cloned now in a much more commercial scale by a more scientific process called tissue culture. Now, this is a sophisticated scientific technique which can produce thousands of clones in a short space of time, and it's known as micropropagation. The way you do it is you find the plant you want with the desired characteristics first. Growers can then just take a few plant cells from the tips of the shoots or the roots. We call these explants. Then they sterilize them to kill any microorganisms, and they place them in a little petri dish containing nutrient medium. They say they are grown in vitro. What we mean by that is in a lab outside the normal biological context. This nutrient medium contains all the growth hormones and the nutrients for the shoots and the roots to start growing. So just from a few tiny cells, you can start to get these tiny little plants growing. The shoots are then removed from those initial explants once they get going, and you can then move those to another petri dish and keep repeating it and repeating it and repeating it and repeating it. So from one original plant, you can end up getting thousands of explants, which then will grow under optimum conditions into full-size plants, which can then be transferred into compost and into greenhouses to develop fully, and they will all be identical to the original plant. Again, this is done all the way around the world in order to maximize the yield of certain crops such as bananas. Now, there's some good things about cloning plants. You get large quantities of plants very quickly. You can grow plants all year round. You can clone sort of rare or endangered plants to increase populations. New plants are grown in sterile conditions, so they're pest and disease free. If you did genetic engineering on a plant first and then cloned it, you wouldn't have to do genetic engineering again, because you've only had to do that complex process once, and then you just clone that one loads of times. However, all the offspring are identical genetically, and that means they are very, very vulnerable to diseases. You also need sterile lab facilities, and they're also really vulnerable to pests.
Is it clear? All clear, Mikhail?
Yes.
Now let's talk about cloning mammals. We just talked about cloning plants by using tissue culture micropropagation. Embryo cloning can be done to clone mammals. It is possible to clone animals using embryo transplants. For example, if a farmer wants the best cattle, they must first create an offspring from the best bull and best cow, and then clone this offspring to create many genetically identical copies. First, choose the best bull, then the best cow, and then clone it. How? This process is known as embryo cloning, and it is achieved in the following way. Step number 1, egg cells from the best cow are artificially fertilized using sperm cells taken from the best bull. So best bull is going to give the sperm cells.
Best daughter or
Is going to give the egg cell. This is going to form an embryo when the sperm cell is going to be fertilized with the egg cell. The developing embryo is then split apart many times before the cells of the embryo become specialized, and this forms many separate embryos that are genetically identical. And then these cloned embryos are transplanted into host mothers. These host mothers, the other cows, we call them surrogate mothers. What is surrogacy?
wait, what?
Surrogate mothers.
Okay.
ठीक है? It's surrogacy, where we take embryos from another cow, plant it into the other host mothers to give birth. The cows born from these host mothers would be all genetically identical as well. So you have taken a cow egg cell, then you have taken a bull's sperm, fertilized it, and you have produced embryos. Then these embryos are split into smaller cells. You can see many identical copies have been made, right? And then they are planted into host mothers. Okay, and those host mothers, which are not the cow and the bull, okay, this is another mother. The cow and the bull was only used to make these embryos and make their copies. Now the copies of these embryos are planted into other cows, which are known as the host mothers. Got it?
Yeah.
And they are then going to produce the calves, and all those calves are going to be genetically identical. It is achieved in the following way. The nucleus is removed from the unfertilized egg cell. The nucleus from an adult body cell, such as a skin cell, is inserted into the egg cell, and a very small electric shock stimulates the egg cell to divide by mitosis to form an embryo. सबसे पहले एक unfertilized egg cell जो आपने cow से ली है, उसमें से you have to remove what? Nucleus. Then the nucleus from an adult body cell, such as the skin cell, is inserted into the egg cell, and electric shock stimulates the egg cell to divide by mitosis, which contains the same genetic information as well. And this same process was used to create the first clone of mammal in 1996 as well, which was done in Scotland, and it produced an adult female sheep that was known as Dolly the sheep. Now what is happening? Female A and female B. Female A, a random body cell has taken from female A. That can be a skin cell, let's say. And from the female B, we have taken the egg cell. Got it?
Yes.
Now, from the egg cell, the nucleus is removed. Okay? The nucleus is removed. And over here, we have extracted a DNA from the स्किन सेल, बिकॉज स्किन सेल्स कंटेन द सेम जेनेटिक इन्फॉर्मेशन। डीएनए हमने निकाल लिए और हमने न्यूक्लियस रिमूव करके डीएनए फ्रॉम शीप ए वाज इंसर्टेड इनटू द एग सेल फ्रॉम शीप बी, बिकॉज वी वांट द प्रॉपर्टीज ऑफ whom, फीमेल ए, राइट। तो हमने फीमेल ए से स्किन सेल लिया, स्किन में से हमने उसका डीएनए एक्सट्रैक्ट किया और डीएनए हमने किसके एग सेल में प्लांट कर दिया, द वन
In
विच वाज
female
टेकन
B.
फ्रॉम फीमेल बी। फीमेल बी का जो एग सेल था उसका अपना डीएनए खत्म, बिकॉज वी हैव रिमूव द होल न्यूक्लियस। ठीक है, न्यूक्लियस
Yeah.
रिमूव हो गया, अब दिस इज एम्प्टी। तो हमने फीमेल ए से जो सिर्फ डीएनए लिया था मॉलिक्यूल, दैट वाज इंसर्टेड इनटू एग सेल फ्रॉम शीप बी, एंड दिस इज हाउ द फर्टिलाइजेशन टुक प्लेस, एम्ब्रियो वाज प्रोड्यूस फीमेल सी। अब ये जो थी दिस वाज फीमेल सी के अंदर हमने इम्प्लांट किया, ए का भी काम नहीं है, बी का भी काम नहीं। नाउ वी फाउंड अनदर मदर शीप विच वाज नोन एज सी, उसके अंदर हमने ये नया प्लांट किया था which contains the egg cell of the bee but the DNA of A, and it was planted where, in C. Got it?
Right. Yeah.
That is how the lamb clone of sheep A was produced. Now let me show you one video as well.
If you already have an animal or plant with all the characteristics you want, then it would be ideal just to make loads and loads of identical copies of it. That is what cloning is, making genetically identical copies of organisms. Cloning animals is a lot harder than plants. You can't just chop off a bit of it, plant it in some soil and it will grow into a clone. Plants have very special areas called meristems which allow them to do that, which are full of stem cells. Animals don't have that. They grow very differently. And it took scientists a bit of time, but in 1996, a sheep called Dolly was the first mammal cloned from an adult cell. The reason, if you want to know why they were called Dolly, was actually, it's a bit of a bad joke by the scientists involved, but it was cloned from a udder cell, which is essentially a mammary gland, and a very well-known country singer, Dolly Parton, has particularly large mammary glands. So they named the sheep Dolly because it came from a mammary gland cell. Anyway, Dolly the sheep, how was Dolly cloned? Well, first a normal adult cell was taken from sheep 1, from the udder of sheep 1 that they wanted to clone. So this was a normal cell with the full number of chromosomes in it, a typical adult cell, just like me taking one of your skin cells, let's say. Okay? Now, the DNA was extracted from that cell because that DNA has the genome in it for that sheep, and that is the entire genetic code needed to clone that sheep. Now, a completely separate sheep, sheep number 2, was taken, and an egg cell was taken out of that sheep. Now, egg cells have the ability to multiply and to grow and form an embryo, so they're very, very important. But you don't want the DNA from that sheep, so that egg cell had its nucleus removed. We call it being enucleated. And the nucleus from sheep 1 was then put in to nucleus from sheep 2. Using electricity, the egg cell is then stimulated to start dividing into an embryo. And then that implanted embryo gets put into a surrogate sheep. It could be a completely different sheep to sheep 1 and sheep 2. So we now got a third sheep involved. And after a while, the embryo grows and the sheep gives birth, and when it gives birth to a clone of sheep 1, because the only DNA involved in the whole process was taken from that initial adult cell of sheep 1. Now that process could be done in exactly the same way with a human, potentially. It hasn't been done, but there's no reason why that method wouldn't work in a human as well. Now, scientists have genetically engineered sheep so that they make medicines for humans in their milk. It's amazing. These medicines can treat things like cystic fibrosis and multiple sclerosis and potentially types of cancer. And the medicines usually come in the form of proteins in the milk, such as antibodies. But when you genetically engineer a sheep and you breed from that sheep, then although that genes you've put in won't necessarily get passed on to the next generation, they might.
So is it all clear?
Yep.
Now you can read these benefits and disadvantages yourself. I'll be back in a few minutes then. Wait.